Okay. Well, thank you everyone for showing up in person and online. Today it's a great pleasure to have at MBI Dr. Scott Echols. Scott Echols is a practicing vet and a research scientist at Texas A&M. He's been at the forefront of developing a lot of novel CT and other imaging agents, and as well as that, tissue preservation, and a lot of groundbreaking work in DICE, which is diffusible iodine contrast enhanced imaging, which we've done quite a bit of at MBI, particularly with Justin and others. So it'll be very exciting to see where that field's going. And he's had a diverse experience across not just lab animals, but it looks like across virtually all of the taxonomies. So looking forward to seeing a really fascinating talk across a whole range of CT disciplines. So thank you, Scott. Welcome to MBI.
Oh, thank you. I'm very excited to be here. It's been beautiful since I've been in town. So I understand you'll had a lot of rain and everything. So I'm very happy to see the wonderful weather and be here. So it's always been an honor for me to come to Australia and to be in this institution as well. So I want to kind of get started and tell you that this is an invitation. This is not just me presenting stuff, because I'm very much into collaborative science, and we're going to see how all this works. And in trying to develop technologies that can be worked and used in a variety of different fields. So kind of a little bit about me. When you look at this, you apparently see, most people see something in it. To me, it's a bunch of dots. And that's all I see. And whatever number or letter or symbol is in this, I cannot see because I'm colorblind. But I also see things a little bit differently in another way too. When I look at your Australian budgie here, I see something that glows. So strangely, I have the ability to see in the ultraviolet spectrum. And why that is, I have no idea. But it's allowed me to kind of look at things in a very different avenue. So if I take this same little budgie and I put a black light on it, that is what I see. Now you can see it now with the ultraviolet, and it's my worthless superpower. So it allows me to see things that glow on some level, and especially birds. And when people have things that glow in their hair, I can see it, but that's about it. But it does mean I tend to look at things a little bit differently. So I have put this up there because I worked with this is a bird food company, Lefevre Vet, and they have offered research opportunities. So if people want to get in touch with me and we want to get on to different programs. So I work with not just veterinarians, but researchers from all different fields of science, and you'll see why in just a moment. This QR code will come up later. So I do develop a lot of this technology that we're going to be seeing. So part of the goal of the research project that we developed was to develop technology, and that's been kind of my field, is how do I solve some of these problems? We're filling in the gaps of what we need to do to accomplish these goals. So this is just one of the alligators, and yes, I've worked on zebras to alligators and kind of everything in between. So let's keep moving here. So what I want to do is describe the comprehensive anatomy research project. And this again is where we develop technology, more with that, and describe some of the technology that we've been developing, and just show some of the projects that we have. And they're certainly not an exhaustive list because we have lots of different projects going on. All right, so the comprehensive anatomy project pretty much covers all animals. And we can even add in, like I said, invertebrates. So we've got insects and spiders in there. We've even dabbled into fungi and plants as well. So the first goal of this project originally started as what was called the Gray Parrot Anatomy Project. And we were focusing on one species. So the idea is that we develop a system of creating standardized nomenclature, and develop kind of a reference for this one species, but it was never about this one bird. It was always about developing a kind of a protocol. And we're still working on the Gray Parrot Anatomy Project, still working on a book, working with anatomists and so forth on that particular subject. The second aim was to develop technologies. And that's again where I come in. I really work to see if we have problems, how do we solve these problems, and what technology is going to be needed to fill that gap to help make these things happen. Third thing is this was never to be a veterinary project. This was always to be a multiple scientific disciplinary project. And that was the goal. And then we bring in computer scientists, we bring in science artists, we bring in, of course, engineers and different people from different aspects of medicine and so forth into this. So that was always a fundamental goal of this particular project. And the last one is to share data, because I can sit on a lot of data, but if I'm just sitting on it and it's not getting out there, it's of no value. So I really want to make sure that we're getting that data. And just in the past few years, I have opened up quite a bit of my data to various researchers openly at conferences, as I'm doing now, and saying, okay, let's see if someone's not using this data, let's use it and do something with it and make most of that, because I've got a lot of data from a variety of different animals, including humans. You'll see a little bit on there. So yes, I do love the technology and the side of this and developing that. It is something that I see as a way to bridge the gap between why we can or cannot see or do certain things. All right. So the first thing I want to discuss are we developed a whole series of CT protocols for live animal imaging. I am a clinician, so I want to take this research information and be able to put it into a live applicable situation where we're dealing with clinical animals. So you can see some of the protocols we developed. Brightview is the first contrast agent I developed, scar tissue stain, and then we'll go in a little bit into the preservation system. So just in terms of protocols, a little bit of history on live contrast imaging is typically based off of human medicine. It started in the 70s and was based off of taking x-rays at a very fast pace and kind of slam in this contrast agent. It's usually iodinated contrast, and it's usually around 2 mils per kilogram. And that got translated over into veterinary medicine, first with cats and dogs, and then it just was everything. And that doesn't always work, especially as the technology is advanced and so forth. So I'm very much into functional imaging. In other words, how do we get most out of this? So we typically take 3 CT scans for every case. That is a plain CT with nothing, a during contrast CT, and then a post-contrast CT. So it gives me functional information about how that contrast is moving through the tissues and lesions that we're seeing, how they're processing contrast and so forth. So in this example, the birds, it's 4 to 6 mils per kilogram, which is considerably higher. But what we're doing is we're giving it over 2 minutes. So instead of slamming it in, which is a typical 10-second protocol using an injector, which gives you a quick spike, and that spike or that peak is where we run into toxicity issues, and then you get a quick drop. Well, if your CT is capturing that data over that very short period of time, you can get a fairly good signal. But what we're doing is capturing the data over a longer period of time. We're not getting as high of a peak, but our data under the curve is larger and we're getting more information over that time. So it allows us to get some really good contrast images, and you're going to see some of that. All right, and this is just an example. This is a bird, and we get a lot of really nice clinical imaging. This particular bird, you can see that right jugular vein is bowed out, and that's because it had a tumor there, and I actually had to use this CT to figure out exactly where the vessel was and carve it out. Reptilian protocol is very similar, 4 to 6 mils per kilogram, and then we sometimes do a second post-contrast CT because reptiles process things a little bit slower. And we use techniques like digital subtraction angiography in clinical practice and looking at flow of vasculature. We do see a lot of cardiovascular disease, not necessarily in reptiles, but certainly in birds. Very, very common. Atherosclerosis is one of the more common problems we run into with birds and collection animals. And it's not just parrots, it's all sorts of birds. So anyways, we are looking at a variety of different techniques to evaluate flow and not just with static CT. All right, our mammal protocol is a little bit lower, and that's 3 to 4 mils per kilogram, but still it's over 2 minutes, and we're not running into toxicity problems that we recognize. So again, it's that functional information I'm interested in. So I'm going to kind of show you an example. So this is a during contrast phase of a dog here. We're seeing the cortex light up very nicely on that kidney. And then if we look at the post-contrast phase, we get the medullary phase, and it's going to go up to the ureters and everything else. So we can look at those 2 different phases. These are very normal kidneys, of course. I can digitally cut this kidney out of the picture And then if I want to, I can look at how that contrast is going through the medullary system. And we can do this on a clinical basis, which is very nice, which allows me to analyze and look at how the function is the kidney, not just from lab work. We know now a lot of our lab work picks up diseases later in the stage. So we really need to look at finding ways to better analyze and evaluate a variety of different diseases, not just kidney problems, before they get to this stage. All right, so BrightView. BrightView is the first contrast agent I developed, and this is 100% non-toxic for the environment, for the handlers. We only use it in terminal cases, so it's not used on living animals. It is compatible with all forms of imaging that we've tried so far. That includes all your X-ray based technologies, CT, MRI, ultrasound, light sheet microscopy, histology. So it works with all these different ones. And it also is a water-based preservation or water-based contrast agent, and it allows us to add in other contrast agents. So we can combine it with MRI agents and light sheet microscopy agents, and it's compatible with that. We've also done multiple different types of stains. On histology, it is compatible as well. So this is just an example here of this golden eagle. There it goes. And the wings are digitally cut off, but you can kind of see it's a whole body contrast. It goes down to the capillary level and crosses over. So we can literally get every vessel in the body. There's a clearing process where we have to get the blood out of the body first. So again, this is a terminal contrast study, but it gives us really beautiful studies, and vascularization allows us to capture the totality of the vascular system, which has been very useful. And you're going to see some of the studies that have come out. We have quite a few studies now that have incorporated BrightView into them to help understand a variety of different diseases. All right, this is one of them. So I'm involved with a lot of neurologic based studies, and this is TBI. My computer is advancing on its own here. So just to kind of, let's go back here. TBI has been a problem because when you look at low-level blast injury studies, whether they're done on rodents, pigs, whatever it may be, we have a very characteristic pattern that the animals go through a blast chamber, they wake up, they're perfectly fine. And it's usually months later, at the earliest, that they start showing problems. And histologically, if you look at their brain, there is no real obvious issue there. So what we did and started working with this group, Mount Sinai in New York, is said, okay, let's look at the microvasculature of the brain. So what you're looking at there in these images, the top images are the control images 8 months after blast, and the bottom 8 months after blast. And you can already see there's significant reduction in blood flow. Volumetrically, there were 3 different analytical systems that were used. It's about a 50 to 60% reduction in blood flow. So it's a significant reduction. Histologically, again, looks normal. So what we're running into is decreased blood flow to the brain. So the group took this a little bit further. Gamma Sosa and his team took these rats out to 18 months. And what you're seeing here, it may look like there's no blood flow, but it's really decreased blood flow. But the interesting thing is they were able to correlate the area of decreased blood flow to the behavior change. So that was the first time we could look at the part of the brain that didn't have a good blood flow because of the TBI and the subsequent behavior abnormality that ensued because of that. The bottom pictures here, these are all normal or controls. And then the team took it out a little bit further, and Gamma Sosa and his team are continuing to work on this. And this is now 13 months, again, a different study, and identifying these microvascular tangles that show up. And now we see microvascular tangles as a result of a number of different types of injury. And and there's just these examples right here. So this is again where BrightView is really helpful, looking at microvascular disease. And I would argue that more and more, a lot of the diseases that we run into are truly a microvascular level. You absolutely have catastrophic large vessel disease, like say when a clot forms and we get a stroke or a heart attack or so forth. However, I would argue that a lot of them start out long before that, and we're getting microvascular changes. All right. In this group, this is actually out of Nature and published 3 or 4 years ago. And this team at Johns Hopkins combined BrightView with galbamin and with rhodamine, and then essentially did micro CT, micro MRI, light sheet microscopy, and histology, and then married or co-registered all those images together to create these really rich, detailed maps of, in this case, the mouse brain. There's the mouse leg on top and the mouse kidney on bottom. So it's another way, again, we've combined BrightView with many different agents and find that it's quite useful to add in other things. Again, being water soluble, it allows us to do this, have this versatility. All right, of course, I work in human medicine as well, and we do a lot with various different human anatomy parts. We're finding so many different anatomical variations, and of course, we expected that, but also we're starting to answer questions we just didn't even know we could find answers to. So there's the same donor, and you can see the BrightView, the rich vascular network that is really showing up there, and then we just kind of remove layer by layer of that, and then we can also see it visually when we do dissections. So this is part of a study done at Virginia Tech on a horse, and we did an entire horse, not the largest we've done so far, but in this case, we've taken the kidney out and performed micro CT on it, and then assigned the outer layer of BrightView, the one voxel, as the only one we're recognizing, and everything else was removed. So it kind of gives you kind of a coating, but it allows you to look at the inside of the vasculature, which we normally look at the vascular tree on the outside, and this is a totally different way to look at it. It looks glossy, like it's computer generated, but it's real data. So again, just another way that we can start to study and evaluate the information that we're given. All right. Again, as someone who teaches, I'm always trying to figure out better ways to educate my student base and the people I'm working with. And I like to look at skulls because they're complex structures, geometrically complex. And when you're teaching somebody looking at, say, an X-ray or it can even be ultrasound, or it can be MRI or CT, understanding what they're looking at is sometimes a challenge with this. So using these models, and they're based off of real data. So this snake has had BrightView, and we did very simple segmentation to kind of segment out the bones, then the vasculature, and a little bit of neuro. It allows us to then create these relatively simple models, but they can be very powerful because you can rotate them any direction you want to match the image that you're looking at. And it can be really helpful for looking at different types of anatomy and trying to understand, is this normal? Is this abnormal? Is there a fracture here, displacement, et cetera? All right, scarlet tissue stain. So this is another stain that I have developed. And what I wanted to do, so this is, you had heard about the DICE CT, diffusible iodine contrast enhanced CT. And definitely have been very much a part of that. This is not iodine contrast. This is something totally different. And this allows us to look at soft tissues, and we can get answers within a day. So we can get answers very quickly and look at detail with that. So I'm just going to kind of show you some examples here. And this is a Pekin duck. So far, this is relatively new. So we only have 2 publications, peer-reviewed publications right now on this. And this is CT, this is micro CT of a duck head. And you can see the really detailed view of the neurologic system, but it also allows us to pick up all soft tissues. So that includes tendons, ligaments, muscles. We've been very interested in seeing the interstitium. We actually did an entire human donor, and we could see the interstitium of the entire body. That was really impressive to see. And there you're actually looking at individual nerves going out to the very tip of the beak on this particular duck. But we've also been looking at lymphatics, and a lot of this stuff is yet to be published, and looking at lymph nodes and everything else. So the stain kind of picks up everything with soft tissue. So it's kind of like the opposite of BrightView. You don't really see the vessels, although you see the lining, the wall of the vessel. So we've actually used it to look at atherosclerosis and vascular disease, but it does everything except the blood vessels, essentially. So it doesn't fill the blood vessels, it leaves them and goes into the soft tissues and allows us to get this kind of detail. So this has really been quite interesting in looking at soft tissue details. All right. So this is just one of our papers that's out. We actually looked at the nerves going to the tip of the beak in a gray parrot, again, part of that gray parrot anatomy study. And this is now published in American Journal of Veterinary Research just a couple of years ago. And we've got more stuff in the works on this. So just kind of show you, this is the brain of a rat and what it's actually staining. So this is the H&E section of the brains stained with the scarlet tissue stain. And it's actually staining white matter tracks in individuals. So we have actually looked at individual nerves and digits and so forth, but that all comes down to your imaging capacity. I think here at the synchrotron, you have that capacity. So you have the ability to get down to these really detailed images and scanning. So that's one of the things I've been very excited about, is actually seeing individual nerves and so forth. And again, we've done this with, we've actually worked it on plants and all sorts of insects and spiders and so forth. So it's provided us some information that we maybe just could not get otherwise. All right. And this picture on the right here is actually a compilation of hummingbirds that were perfused with Brightview and my garden plants that have the scarlet tissue stain, and I added false color to it. And it ended up on the cover of an album for a musician out of Europe. So anyways, it was kind of fun to have that. I guess that's one of the, when you've made it in science, is you actually make it on the cover of a rock album. That's it. Yeah. So, all right. Okay, now onto the Scarlet Imaging Animal Preservation System. This is one of the things we were working on yesterday. So I wanted to create a preservation system that, first of all, has no formaldehyde. I guess I'm a little shocked that we're still using this worldwide. It's nothing in the States or here in Australia, it's everywhere. But I wanted to get away from the common preservation chemicals. That's phenol, formaldehyde, ethanol, salt solutions. And I wanted it to be able to mimic soft tissue characteristics that are realistic. I'm a surgeon. I'm specifically a bird microsurgeon, but I do surgery on all sorts of animals. And I wanted to be able to teach people how to do these procedures without putting any animals at risk. I also had to make sure it's compatible with Brightview. I also had to make sure that it's compatible with histology. And I also want to be able to pump real blood or artificial blood, however I want to do that. So this is a very nervous student doing a pancreatic biopsy on a pigeon. And this pigeon has been deceased for months. But the pigeon, you can see the tissue looks very lifelike. And what we've done is in this case, we're using artificial blood. And I think at some point I was telling the student, okay, you can't do it this way. This is clearly not working. So, but the good news is it's okay to be nervous. It's okay to be shaky, and you're not going to hurt anything. Let's get the technique down. Let's work on figuring out how we're going to do this. So let me show you how to handle this here. So let's do this in such a way that we're not going to damage the tissue on a living animal. And it allows us to practice these types of procedures. Now, we have done these procedures all the way up to brain surgery in humans. In order to be able to do brain surgery, we have to preserve the tissues in a way that allows us to get very fresh tissues for neurologic tissues. But if we're talking about muscles and most organs, we don't have to have them as fresh. And we have certainly worked with a number of donors, whether it be human or animal, that are months or years in the freezer. They're not as ideal, but we're still able to use those tissues. So this is an example of human surgery. This is robotic adrenalectomy. And what you're looking at the top here, this is the diaphragm. What you're going to notice as we go through this is the diaphragm is moving. And that is because this person has been intubated and they're actually providing intermittent positive pressure ventilation. Now, the white that you're seeing is Brightview in the vessels. You can see that the Brightview is very flexible and pliable, and it's radio dense. So it allows us to do CT imaging, but it's also visually very easy to identify. And in this case, I'm going to go through here and just kind of advance here. So this is the adrenal gland right here, and this is the vena cava. And again, I want to show you here, there's the breathing that you can see. Okay, so in this particular model, this is a non-bleeding model. Okay, in this case, because the Brightview has filled the blood vessels, we can't add anything more, but we could do it without any contrast, and then we can inject contrast into that. So this particular surgeon did something that actually changed how this particular institution started teaching these surgeries. So she is here very delicately cutting the adrenal gland away and lifting it off the vena cava. And she said, Scott, I want you to come over here and look at something. I said, okay. She said, you see, I'm pulling this adrenal gland off. I'm doing this by the book. I am following all the guidelines. I said, yeah, it's, very careful procedure, and you've got the Da Vinci robotic system, everything looks great. She says, well, let's change this whole situation. She said, we teach students how to do things the correct way, which is ideal. What happens when they mess up? And she says, watch this. I'm going to cut the adrenal vein, which is what she's cutting, which leads directly into the vena cava. It's a common site where complications occur. And I said, well, that's a bad complication, cutting a hole that leads directly to the vena cava. And she says, you're absolutely right. Most people die if they're not dealt with immediately. So she then calls over her lead, her senior resident, and says, Dr. Smith, you just cut the adrenal vein. You have 2 minutes to save your patient. Go. And he had to jump into the chair and start working in time. She said, we really need to be teaching people how to deal with the mishaps and the other problems, because not everybody follows the book, and the anatomy is highly variable. And that particular situation has expanded in this particular institution, and they are continuing to do that. This is another thing we've been working on. You're just seeing some preliminary work here. These are red and blue bright view. It stops at the capillary level and allows us to get one system or the other. So we can do just the arterial system, CT scan, and then we can add in the blue and do the venous system and CT scan. So, or we can do it the other way around, or just one or the other. So this is something we've also been working on, and that is to have contrast that just fills one vascular system, but not the other. And we've been working on that. So this is actually if I remember correctly, a vulture, and just looking at, no, it's not a vulture, it's one of our parrots, and looking at the underside of the wing there and the vasculature. So these are just some of the new things to come. And this is looking at the turkey vulture brain, and you can see the red arterial portions of the brain and the blue venous portions of the brain. So we are doing this, and we've done this with humans, we've done it with rodents, and now with a number of different bird species. All right. So that's some of the technology. I want to show you just the projects. We have lots of projects. I'm not going to go through all of them. And the goal here is not to describe everything we're doing, just to kind of give you a flavor of what's going on. So these are just a couple of projects: the penguin anatomy project, tortoise anatomy, rhino anatomy. So the penguin anatomy project is being conducted primarily at Midwestern University, which is in Glendale, Arizona. And it's part of the archives study, and it's something that we're continuing on. We've had 3 penguins entered into the project, and we're obviously looking for more as they come. So the way we get animals is that these animals have to be euthanized. So we will not go and get animals to euthanize them. They are determined that it's best interest of the animal by the animal care team and the owners and so forth, that this animal should be euthanized. And then they're entered in this study. We have forms signed so that we can work with various universities. So we have a track record of everything. And then the animals are humanely euthanized under anesthesia, or in this case by the facility, and then the animal is shipped to us. This may sound strange, but we get some of the weirdest animals in the mail. And we've had a couple snow leopards show up. We've had octopus. We had one day where we had 10 live bamboo sharks show up. You just never know what's going to come through the door. I think my poor staff is wondering what's in the box. So it's kind of fun to see that. But we have already identified, and we have one full publication out now, just came out earlier this year. And we have 2 macaroni, 1 Adelie penguin, and of course we're looking for more. And this is just part of one of the images from the study that just got published. So we are continuing to work on this and looking for more animals to enter into the studies. We also have a lot of other bird projects. Of course, we have the Great Parrot Anatomy Project. We've been working on the respiratory system. Got a Nature paper out of that just in the past year. And that was looking at the subpectoral diverticuli of soaring birds. And we showed that the birds can actually control and manipulate that to help us soaring. And that's what made the Nature paper and made the cover as well. We have the Australian birds anatomy. So we have a lot of people interested in nectar feeding birds and some of the others. Domestic duck, emu, which is different from the Australian Birds Anatomy Project, and then the sea turtle anatomy, and so many more. We have lots of different studies come out. So I should bring up how we're paying for this. How in the world is this even happening? It's really through donations. So we have quite a few different individuals and companies that have contributed to these projects. And what I try to do is work through my hospital and make them as inexpensive as possible. And then we also do research for a lot of universities. So we'll get data collected for them and either send it to them digitally or physically. Sometimes we send the animal back to them so they can do some things. We can do things far quicker and more efficiently by doing them through the hospital. And we have all the protocols and everything written down, so we make sure we follow the various guidelines that are needed for those animals and those facilities as well. All right, so the tortoise anatomy project is also being done by a different group led by Heather Smith at Midwestern University in Glendale, Arizona. I think you're familiar with the anatomical record. She is the editor-in-chief in the anatomical record. She is also well known in paleobiology and working with turtle specimens, ancient extinct turtle specimens. And she's oftentimes digging in Utah and other places and finding all sorts of crazy creatures out in the desert. And I'm the other project director working on this particular project. So we have been fortunate. We have a lot of veterinary staff and students and so forth that get to be a part of this project. And again, this has been a great experience, especially for young up and coming people who want to get involved in research, maybe don't exactly know how, and we can kind of slide them into various projects. And we have lots of projects going on and get them involved on data analysis and so forth, or they can be directly involved with dissection, or they can be learning how to do segmentation. There's lots of different skills that they can learn while going through this process. So why tortoises? Well, turtles and tortoise species are the most endangered vertebrate class of animals. So they're at high risk of extinction or in reduced numbers worldwide. So we don't have that much information on them. And they're commonly kept as pets, which is good for us, which means we have access to get animals that have to be euthanized over time, and we're able to use them into the different studies. So we want to accurately document their anatomic features. We want to relate those into clinically useful information and, of course, publish this finding and build upon this and help with conservation of these animals. We've collected quite a few tortoises to date, and we're working on them, and we do a variety of different studies that come in. So I'll kind of go over that. Some of them, we get live cases. So I'm always shocked at what people have as pets, at least in the States. I'm like, what are you doing with this highly illegal animal? And they're like, oh, I bought it at the pet store. I'm like, okay, I can't help that, but and so animal trafficking is still quite prevalent, including the reptile trade, insects, fish. These are all big areas of animal trafficking, and they are worldwide. So of course, these animals are being taken from the wild, and we don't know that much about them. So we do several things. If they're a live animal and they're clinical, I end up paying for the scan. I just do it. I say, let's just get some x-rays and CT imaging just so we have some baseline information. We're not doing this for medical purposes. We're just, and a lot of times the owners are absolutely, they recognize they've got an unusual animal, and we want to try to use that animal for information. Nice thing about tortoises, they just sit there when we do a CT scan, so we don't really have to anesthetize them or anything like that. And they're usually pretty willing participants. Now, if we have them that come in for euthanasia, this is our typical pattern with most animals, whether it's a bird or reptile or something else, is we put them under anesthesia. So it's a terminal process. They never wake up. And what we do is we'll first CT scan them. Sometimes we get x-rays and everything else, so we have some baseline information. Sometimes we'll get blood work. We do a CT scan. We go through our typical CT protocol. And what I do is I modify that protocol just about every time, because I'm trying to try different values. Again, these animals are getting euthanized, so we basically are using this information that can be applied later to clinical cases. They then, after we do this full CT protocol, remember we're going to do a pre, during, and post-contrast study. Then we'll euthanize them under anesthesia. We'll preserve them and use the preservation system. And then from there, we will go in and submit them for more CT imaging, sometimes even take them to micro CT. And then we farm them out to the various researchers who can then do more detailed dissection, anatomy discovery, etc. All right, so we oftentimes get weight, physical exam, size. We're collecting all the data we can. we have this whole system that I just described of how we do this whole flow that's very typical with each of our subjects that get into the program. And again, it's not just a turtle, but we wanted to focus on 5 areas of research for our tortoise species: vascular anatomy, joints and orthopedics, respiratory anatomy, shell anatomy, and advanced imaging atlas development. So again, we have donors for this as well. We have people who are very interested in seeing these things happen, and that's what helps me allow to do these things. I can't, we looked at the grant writing system, and it's very difficult. A lot of people are not interested in providing money for tortoises, for example, on the federal level. And the other problem is that I can't tell you when one of these animals is going to come in. And so for me to write a grant and wait for that to happen, we just said, let's just do it and we'll figure this out later. And we have donors that kind of help fill in those gaps. All right, so we generate these detailed vascular maps. You can see this turtle has a prolapse here, and the intestines have prolapsed, and it was chronic, which is the reason that it was ultimately euthanized. And but we go ahead and take all the information we can possibly get from every animal. My goal and my philosophy is that if someone has donated their animal, whether it's a pet or not, I'm going to do my very best to find the absolute most I can about this animal and involve as many people to dig through this data. Of course, once we have the data, we can do all sorts of things with it, and it can be done years later too. So the collection of data is quite important and allows us to then look at this in a more detailed way later as needed. So this is just an MIP here, looking at the vascular nap. You can see the heart here in the central. We have a couple publications and works on just that. And this is some of the anatomy that's been developed looking at a variety of different species of tortoises. This is the forelimb here. And the reason I point this out, in veterinary medicine, we commonly stick a needle into this region thinking we're hitting a sinus. Well, what we're doing is spearing a capillary and vascular network and shearing it and damaging it and collecting the blood that is pooling. But we didn't know that until we did this. We had no idea. There is no sinus there. It's just a bunch of vessels. We see problems with joint problems, with orthopedics, with turtles, and we really don't have a good handle on how to deal with those. So we've been looking at the myology and the joints and limbs and everything else. And again, not trying to get into all the specifics. This is an example where we have a dislocated hip. You can see the hip here on the right is ball and socket, and you look here on the left, it's completely ventrally dislocated. And you know, we've used these anatomy studies to figure out, okay, how do we actually approach this without causing great damage? How do we solve this problem? And we actually did it with this particular tortoise. Respiratory anatomy is another area that, again, we've kind of decided to work on, and we have publications in works for those, and looking at all the different anatomic features and looking at the variety of tortoises that we have, their interesting adaptations, and their environmental adaptations and so forth. And they're quite interesting animals the more we look at it. Shell anatomy is important because there's a lot of road traffic accidents. And I'm sure you have the same thing here as there are seem to be in every country, where we have these injuries, and can we save these animals? You know, and road injuries are quite common. So we're looking at shell anatomy to try to figure out where is it best to repair, how we can best repair these animals. And then again, of course, creating those atlases are going to be ultimately important for everyday use of for veterinarians, especially in those who are in the care of these animals. And this is just an example of live animal imaging. We now have these protocols down that we can routinely have animals come in. We have a protocol for tortoises and everything that has come out of this. So when we need to do CT imaging, we have a good idea as to where to begin. This again is digital subtraction angiography. And it's kind of interesting to see how the blood kind of slowly goes through these tortoises and turtles. And this is a box turtle, but very similar, as slowly is distributed throughout the body. So this allows us room to look at, if we want to consider physiology studies and other basic science questions that need to be asked. So it can be everything, again, from that type of research to clinical and everything in between. All right, we've done MRI. There's a little brain here of this yellowfoot tortoise. There's another view right here. So we're exploring different ways to do that. So future directions, we absolutely are working on getting these publications out. The goal is ultimately to get this in the literature so it can be used, it can be scrutinized, it can be built upon, and we need to get that. We're still working on a variety of donations. I really am a huge fan of getting students involved in science so they see it, especially earlier on their career, so it at least is on their radar as an option for them in the future. And incorporation into primary education is another thing we're doing. We're actually bringing in these types of scans and studies into high school classrooms through VR, so they can actually study these animals while in their classroom. All right, and here's some more studies we have. We have the green iguana, we have Mojave rattlesnake, monitor anatomy, sea turtle, which we mentioned again, tortoise anatomy. So there's a lot of different studies going on. All right, this is one of the biggest ones, physically big and project-wise large. So Mitzi Klein, who was here at ANSTO, was very much involved in actually instigating this whole project. When she called me and says, "What do you think about doing a rhino?" I'm like, how do we do that? Trying to figure that out was quite a chore. And we had this international coalition of people from Australia, South Africa, throughout the United States. And ultimately I said, look, there are rhinos in captivity. And unfortunately, they pass away. Let's get everything in place, have agreed upon how we're going to do this. And when the day comes, we'll be ready. Well, I got a phone call. We have a rhino. And you never know when that day will come. And sure enough, we had to euthanize an elderly rhino. And it happened to be in my backyard in Salt Lake City, which made things very convenient because you can imagine shipping chemicals and everything for a rhino is not an easy task, but it made it a lot easier. It was just a local delivery as opposed to a long haul delivery. So lots of people involved in this. And yes, we have done that. The main concern is the rhino horn trade, and that is why they're being decimated, especially the black and white rhino. And it is a continuing problem that does not seem to be resolving. So we wanted to look at that. So we have a lot of rhinoceroses in captivity, of which we have very little information on. And the anatomy information we have, as we're finding, is not very accurate. And at a meeting here in Melbourne, just in a few weeks, a lot of these studies will be presented. I think right now there's roughly 25 presentations and just on this one rhino at that meeting. And most of it will be in the form of posters and some presentations. So there's a gigantic rhino aorta. Everything is large size, of course, and trying to figure out. So what we did is every tissue, we couldn't run a rhino through a CT, it's a little too big to just physically pick up. So we had to do this particular rhino in pieces, but we did CT and MRI of all the tissues prior to doing anything. Then we did the preservation, then we did bright view, even added in gadolinium. And then we did CT and MRI again. And then the different tissues got distributed to the various researchers who were allocated those tissues. And they are currently working on dissecting them and going through them. So it's been quite a process. And I think this year we'll finally start to see some of the data coming out. That was December 2024 when we started this. So, everything takes time, but we're getting there. So there's some of the liver, just a single lobe, and everything was gigantic size with this. And it was a physically demanding experience. And I don't think I can ever forget walking into the belly of a rhinoceros and being able to sit inside this gigantic cavity. It's just amazing. And unfortunately, this rhinoceros had cancer. So it was definitely time. But again, as the philosophy with all these animals, we're going to do everything we can to get the most out of this information. All right, so we also figured out we did the technique later with the separating out the arteries from the veins and the kidneys of the rhino. That worked really well. So that was nice. We had two kidneys, so we did arteries first and then veins on one kidney, and then veins first and arteries on the other, and got some beautiful images from that. And of course, Lefevre Company has helped with a lot of the funding on this particular project. All right, and more mammal studies. We have the Ferret Anatomy Project, the snow leopard, which we have publications on now, Australian marsupial anatomic study, California sea lion study, sloth study. So this is just kind of give you an idea. There's a lot of projects that are going on. We have, I have a giant Pacific octopus at the hospital waiting to find a home. We have a cane toad study, which you guys have problems with. We do as well. They're now invasive all over Florida and some parts of the Southeast. We have the bamboo shark that I mentioned, various insects, plants, fungi. So we're using this technology to look into multiple different fields. So again, if people are interested in getting involved, there's the QR code and they can go to the Lefebvre website, and that's how they will ultimately get in touch with me. I'll provide my email here in just a moment as well. But we want students to get involved and get interested and excited about science, and hopefully find some passions that maybe they didn't even know they had. So thank you for your time. I think I have another 10 minutes or so. If there are any questions, I'm more than happy to answer those questions. So yeah, please let me know.
Yep, thank you. Awesome. Wow. Thank you, Scott. That's fascinating stuff. My gosh, so many insides of creatures. Has anyone got any questions from the audience? We don't have any online.
Thank you for your great work. I'm Peggy.